In the previous video, we looked at site-specific recombination events. Now we're going to look at homologous recombination, which involves identical or nearly identical stretches of DNA that can undergo recombination. And I want to draw your attention to a couple of videos that I think very accurately illustrate what happens during the formation of what's called a holiday structure during homologous recombination. Homologous recombination is important for an organism because it can be used to repair damaged regions of DNA. It can also be used to defend against foreign DNA, or in some cases incorporate foreign DNA if there's relatedness in the DNA. And finally, this really is important because it contributes greatly to genetic diversity. Here's a list of some of the protein components that are involved in this, protein and gene components. So we'll be looking at RecA, which binds single-stranded DNA, the RecBCD complex, and the RUV proteins that are involved in branch migration. We will not talk about the pre-proteins, and as we've already learned from previous lectures, DNA polymerase and ligase will be important because they play a role, as they always do, in filling in gaps in DNA and sealing the backbone. And finally, there's a chi sequence that we'll be talking about. Robin Holliday proposed a model to explain how recombination could give rise to two different types of products. And in his model, he invoked something called the cruciform structure. So we're going to look at how that works. So first of all, if you look at the very left, here is our event in which we've seen some strand invasion. So part of the strand that was associated with the black DNA molecule has now had a nick, and it's invaded the lower strand of DNA, or the second strand, and the corresponding region from the blue DNA has migrated up to base pair with the DNA on the top. And remember that we've got homologous DNA here, so we'll have base pairing. Now what Holliday proposed is that if we look at how the DNAs are arranged in this configuration, first we can see a structure that looks like this. One, and that's just looking at the crossover event here. We're putting that in the middle, and the DNA up here is shown here. This part of the DNA on the other side of that cross is shown down here, etc. We're just looking at it in a slightly different orientation. When you get this structure forming, the RUV proteins, which we'll talk about in a minute, can recognize this structure and can cut the DNA at that position. If you have that type of resolution, then we get what's called a patch. And the way that that happens is that you want to look at the cutting occurring there so that you get this piece of DNA up here, and the other strand of DNA that's liberated is shown down here. And notice that we've only had a small region of the DNA from the blue strand that has joined with the capital A, capital B strand, and conversely the same region of DNA that was originally on the black double-stranded DNA molecule is shown now with the blue DNA. So this is called a patch because it's a very short region that has been exchanged. The DNA can also undergo an isomerization. So again, let's look at this structure that has resulted from the crossover. Now if we rotate this 180 degrees, we can see that here we have this part of the DNA now is on the left side, and this piece is now on the right side. And this cruciform structure can undergo a second isomerization. And in this case, now we're going to rotate along this plane. And now you see that the DNA that was on the top here, oops, is now on the bottom, and this piece of DNA is now on the top. Now when the RUV proteins bind to this sequence, we get cutting in this plane. Notice over here it was in this direction. We're now seeing cutting here so that we liberate these two DNA molecules. And we've got this one, oops, this one down here. And this one is shown here. And this gives us complete exchange of flanking markers. Some authors call it a splice, but you'll see that now we have lowercase a paired with capital B and capital A paired with lowercase b. So just to recap, we've got our crossover occurring here. That's our starting material. We can have resolution, or we can have an isomerization here to give us this structure, a second isomerization in this plane to give us structure B, and when that resolves we get a different product. When crossing over occurs, again, so let's look at the regions where a little bit of crossing over has occurred. Something called branch migration can occur. And we call it branch migration of the holiday junction because, as you'll see, it's actually this cruciform structure that is going to move in this direction. And of course, a consequence here, and the reason we were talking about why this is important for genetic diversity, is that we've got two molecules of DNA, the blue and the black here, that are virtually identical, but they're not completely identical. There's a GC pair here, and in the corresponding region on the other strand of DNA we have an AT pair. So in our recombination event that's going to occur, we're going to have exchange of these. And that's shown here. Now we've seen that this holiday junction has migrated in this direction, and now we have a GA pair up here which is not going to work. That's going to undergo some repair, and we don't know which base will win, and the same thing is true here. The proteins that are required for this, and we'll come back to these in a few minutes, are the RecA, B, and C complex. Well, let's look at the proteins that initiate this recombination event. So first of all, we're going to look at the RecBCD trimer protein complex that can bind to a double-stranded break, and it both separates the DNA because it has helicase activity. So we'll see it moving in here. You can see the DNA is separated here, but it also degrades the DNA because it has exonuclease activity. Now, the first thing that it does is it's going to degrade the DNA from the 3' end, in this case, until it reaches a chi site. And here is a chi site, or an example of a chi site in many enteric bacteria, but it's going to be slightly different in different organisms. And when it reaches the chi site, the 3' to 5' exonuclease activity slows down, and instead the 5' to 3' nuclease activity increases. So again, first it has a 3' to 5' activity. It reaches the chi site, and then it switches from 5' to 3', meaning it will be able to degrade the lower strand. Well, what this does is it allows the creation of an overhang. And this is the overhang shown here. And we have RecA bound to it. So once it becomes this single-stranded molecule, we have the entry of RecA into this complex. And then we're going to look at how that complex, shown in C, is able to create what's called the D loop, shown down here. And this is the formation of the structure that now becomes our holiday junction. So we're going to flip this. Notice that instead of being 5' to 3' orientation here, we've got the showing the 5' end on the bottom here. And that now can invade and find a homologous region on this other strand of DNA. The RecA protein carries out many functions in this process. First, it's going to be able to bind to single-stranded DNA from any type of a break in DNA. And when it does that, it doesn't require energy to do that, but it can then form what's called a 3-stranded structure where the RecA bound single-stranded DNA can invade double-stranded DNA where there's homology. And then you get basically swapping of one strand of DNA for another, and that's shown here. You can see that there's a yellow and a red strand, and we are now displacing one of the strands of DNA. So again, we're generating some genetic diversity when we do this. And that's shown a little bit in greater detail in this figure that is from your book. So we've got, here's our DNA, single-stranded DNA that's got RecA bound to it, and we're moving onto the DNA, the spooling in of the double-stranded DNA, and at the other end we can see that now our DNA that was coded by RecA is now the DNA that's paired with the double-stranded DNA that we started with, and here is the displaced strand of DNA. Well, finally, the RUV proteins are going to help to resolve these structures, the cruciforms that form during homologous recombination. So first of all, RUV-A is a tetramer, forms a tetramer, 4 subunits of the same protein that will bind to this cruciform structure shown here. And the RUV-B protein can recognize that complex, and you'll see that there are various configurations. You can have 1 or 2 of the RUV-A tetramers. RUV-B, by the way, is a hexameric structure, meaning there are 6 subunits of RUV-B that wrap around this. And the arrows indicate that in this figure that when these proteins are bound, we can continue to see some branch migration of this. But finally, the RUV-C protein will be able to bind to this complex. And RUV-C, a homodimer, is called a cruciform cutting endonuclease. In other words, it will be the molecule, the protein that binds to this and cuts the DNA, and you can see that here. And when we have that endonuclease activity, we end up with little a being joined to big B. Notice up here it was capital A, capital B, little a, little b, and now we have B is joined to, little b is joined to capital A, so we've had a homologous recombination event. And finally, ligase will come in and seal the 5' and 3' ends. Well, this is an example of something called recombineering. All right, well, that doesn't look great, but recombineering, which really takes some of the enzymes that are important for homologous recombination and uses them to generate new molecular tools. So in this case, here's a target, perhaps you want to make an exchange on the chromosome, and what you do is look at the DNA that flanks that target. So let's say that this is the RecZ gene, and we want to replace it with a marker, GFP or kanamyosin resistance, whatever you want. These regions that flank the RecZ gene can be very small as 50 base pairs. And you construct this as your replacement for that. So you match those 50 base pairs on the end, you engineer your sequence to contain those, and then put your marker in between, your GFP or kanamyosin resistance gene. You introduce this construct into the cell along with something called PRED. And what this is a construct that contains 2 lambda phage proteins that will mediate recombination between this marker with the homologous regions at the ends with your target on the chromosome, so that you end up with a very clean construct on the chromosome. It's very efficient and very inexpensive, and there's virtually no size limit to what you can do, and it saves a lot of time. Here's another example of recombineering, but in this case, instead of having the lambda red genes on a plasmid, they're actually on the chromosome. So here we've got a lambda prophage, a virus that's integrated onto the chromosome, and we've got 3 genes of interest: EXO, which is an exonuclease, BET, which will encode a RecA-like protein, and GAM, which will encode an inhibitor of the host RecBCD proteins, because remember, RecBCD, that complex of proteins, will degrade DNA, and we don't want to do that during this operation. So the other thing that's important to note is that there's a lambda promoter, and this lambda promoter is heat inducible. So that's going to allow us to turn on expression of these genes at a very particular time. We'll use heat to turn on expression. So we're going to use this E. coli strain to engineer some changes to a gene on a plasmid. So let's look down here. Here's the target plasmid, and we've got it in the cell, and we would like to change a gene on that plasmid. We would like to replace it, in this example, with an antibiotic resistance gene. So let's go back up to here and look at how we're going to do that. So remember, we want homology. So this gene down here shown in green, we have some sequence information. We know what the flanking DNA is, so I'm going to draw that out here. Here's our gene X that we want to replace. We've got DNA that flanks that, and I'm going to call them A and B. We're going to take that information and use the sequence of A to make a region, a PCR primer. So that's here. And we're going to take the sequence of B and use it to make a second PCR primer. Those PCR primers will be designed in such a way that they not only have homology to A and B, but they also allow us to amplify this antibiotic resistance cassette. So we generate a PCR product. It's very simple. And here we've got in blue, our antibiotic resistance gene, and at one end is DNA that's homologous to region A, and at the other end we've got DNA homologous to region B. Now we heat our cells, and that's going to induce expression of those lambda, EXO, BET, and GAM genes. We're going to electroporate this PCR product into the cell. Now when we electroporate in, we've already induced expression of red. So we have the red proteins being made, EXO, BET, and GAM. And our regions of homology will have 2 recombination events at either end. And what that's going to do is when we recombine those, we're going to delete this gene and replace it with that antibiotic resistance gene that we were interested in. So here we end up with our final product. On the plasmid, we've taken out the gene that we were wanting to replace, and we've put in an antibiotic resistance gene instead. Well, let's look at another example. And in this case, they're actually using single-stranded oligonucleotides to make a replacement. So here let's look at example number 2, but a deletion would be done in the same way. We synthesize an oligonucleotide, and it's going to match our gene of interest down here on the plasmid, but we've added a single base pair change. Now we again induce the lambda red proteins. We electroporate that oligonucleotide into the cell, so you can see it here. And because we've got homology, we will again delete or remove the gene that was there, and we will now replace it with the gene that has our point mutation of interest on the plasmid.